Hello everyone and welcome to today's webinar titled Automation of the Pointe Scientific Glucose 6-Phosphate Dehydrogenase Assay for High-Throughput Use in the Clinical Laboratory. Today's webinar will allow viewers to earn 1 Accent Continuing Education credit by completing the evaluation form at the end of the webinar. Today's webinar will be hosted by Dr. Erin Milner, who currently serves as an assistant professor at the United States Military Academy in West Point, New York. Prior to this, Dr. Milner served as a Chief of Laboratory Services at Eisenhower Army Medical Center. Dr. Milner holds a Bachelor's of Science in Chemistry and Mathematics from Kennesaw State University, a PhD in Chemistry from the University of North Carolina at Chapel Hill, and has also earned her Diplomat of the American Board of Clinical Pharmacology Board certification. If you have any questions during the webinar, please submit them utilizing the live chat feature, and Dr. Milner will try to respond to them all by the end of the webinar. If she cannot get to all the questions, please feel free to email her at erin.emilner.mil@mail.mil. It's shown on the screen now. At this time, I would like to welcome Dr. Erin Milner.
Thank you so much. I appreciate that introduction. So moving on to the second slide, if you would like detailed methods information, I've listed our publication. Also, please note that MedTest DX is sponsoring this American Association of Clinical Chemists webinar. Also, as a disclosure statement, neither I nor any member of my immediate family has a financial relationship with the company as defined in the AACC policy on disclosure of potential bias or conflict of interest. Another disclaimer, the opinions or assertions contained herein are the private views of the author and are not to be construed as official or as reflecting the views of the United States Military Academy, Eisenhower Army Medical Center, the Department of the Army, or the Department of Defense. So moving on to our agenda, I have listed here. We're going to get initiated with the epidemiology associated with G6PD and the clinical manifestations. Okay, an estimated 400 million people worldwide have glucose 6-phosphate dehydrogenase deficiency. This condition occurs most frequently in certain parts of Africa, Asia, the Mediterranean, and the Middle East. It affects 1 in 10 African American males in the United States as well. The G6PD gene provides the code for ribosomes to construct the enzyme glucose 6-phosphate dehydrogenase. There are more than 200 mutations that cause G6PD deficiency, and they've been identified associated with the G6PD gene. Although all of these mutations lead to changes with respect to particular amino acids in the G6PD enzyme, these changes disrupt the normal structure and function of the enzyme or reduce the amount of the enzyme produced in cells. It should be noted this condition is inherited as an X-linked recessive pattern. Therefore, in males, 1 altered copy of the X-linked gene is sufficient to cause the condition. In females, the mutation would have to occur in both copies of the gene to cause the recessive disorder. Subsequently, males are affected more frequently than female. Now let's adjust to discussing G6PD pharmacology. The G6PD enzyme, which is active in virtually all types of cells, is involved in normal processing of carbohydrates. It plays a critical role in red blood cells, which carry oxygen from the lungs to tissues throughout the body. This enzyme helps protect red blood cells from damage and premature destruction. G6PD is responsible for the first step in the pentose phosphate pathway, a series of chemical reactions that convert glucose to ribose 5-phosphate. Now, ribose 5-phosphate is an important component of nucleotides, which, as we know, are the building blocks of DNA and RNA. This chemical reaction produces a molecule called NADPH, which plays a critical role in protecting cells from potentially harmful molecules called reactive oxygen species. These molecules are byproducts of normal cellular functions. Reactions involving NADPH produce compounds to prevent reactive oxygen species from reaching toxic concentrations within cells. The production of NADPH by G6PD is essential in red blood cells, which are particularly susceptible to damage by reactive oxygen species because they lack other NADPH producing enzymes. Okay, so the G6PD gene exhibits a high amount of polymorphism, resulting in a range of G6PD activity from normal to severely deficient. Variants are classified 1 through 5 by the amount of activity of the G6PD enzyme, as indicated in the World Health Organization classifications. While a low level of G6PD enzyme indicates a deficiency, an affected person is more likely to experience symptoms when exposed to a trigger. Therefore, the severity of symptoms will vary from individual to individual and from episode to episode. A normal G6PD enzyme level in a male indicates that it is unlikely he has a deficiency. And if anemia is present, it's likely due to another cause. However, if the test was performed during an episode of hemolytic anemia, it should be repeated several weeks later when the RBC population has had time to replenish and mature. Now, women are carriers, having one mutated and one normal gene copy, right, also known as a heterozygous carrier. They'll have some RBCs that are G6PD deficient and some that are not. These women will usually have normal or near normal G6PD levels and rarely experience symptoms. Therefore, a carrier may not be identified through G6PD screening, but would be detected in a G6PD confirmation test that quantifies the overall amount of enzyme present in the cells. Note that rare female who has two mutated genes, who would be homozygous, will likely show a significant decrease in G6PD level. Now, for the clinical manifestations, without adequate functional G6PD, red blood cells are unable to protect themselves from the damaging effects of reactive oxygen species. The damaged cells are likely to hemolyze, and that hemolytic anemia occurs when red blood cells are destroyed faster than the body can replace them. This type of anemia leads to paleness, yellowing of the skin, whiteness of the eyes, also known as jaundice, dark urine, fatigue, shortness of breath, and a rapid heart rate. In people with G6PD deficiency, hemolytic anemia is most often triggered by bacterial or viral infections or by certain drugs, such as some antibiotics and medications used to treat malaria. Hemolytic anemia can also occur after eating fava beans or inhaling pollen from fava plants, and that reaction is called favism. G6PD deficiency is also a significant cause of mild to severe jaundice in newborns. Many people with the disorder, however, never experience any signs or symptoms and are unaware they have the condition, which is why G6PD screening is important. Now, G6PD screening is often ordered with other laboratory tests consistent with hemolytic anemia. Clinicians will be seeking information relative to increased bilirubin concentrations, hemoglobin in the urine, decreased RBC counts, and haptoglobin levels, increased reticulocyte count and lactate dehydrogenase levels, the presence of bite cells on a blood smear, and sometimes the presence of Heinz bodies inside the RBCs on a specialty stained smear. Screening may be performed on a newborn in the first day or two after birth, if recommended or required prior to drug dosing, or through the Soldier Readiness Program. Now, regarding drug-induced hemolysis, patients should be tested for G6PD deficiency before being prescribed primaquine and tafenoquine for radical cure, terminal prophylaxis, or malaria chemoprophylaxis due to known liabilities associated with hemolytic reactions in patients with deficiencies. The FDA contraindicates drugs with predictable hemolysis in patients with G6PD deficiency or unknown G6PD status due to the risk of hemolytic anemia. This potential liability has led to mandatory G6PD testing for service members to ensure deployment readiness. Increasingly, the relationship between primaquine, G6PD, and Plasmodium falciparum is intriguing in that P. falciparum has been a major selective force in increasing the frequency of G6PD deficiency. Researchers believe that people who have a G6PD mutation may be partially protected against malaria, and a reduction in the amount of functional G6PD appears to make it more difficult for the parasite to invade red blood cells. G6PD deficiency, therefore, occurs most frequently in areas of the world where malaria is common. Now, from our perspective, in terms of the G6PD testing options, we performed a survey of commercially available G6PD assays for high-throughput screening in the clinical laboratory. And that yielded two viable vendors, Pointe Scientific and Trinity Biotech. Now, while at Eisenhower Army Medical Center, we had previously implemented the Trinity Biotech method. We were informed that manufacturing would discontinue beyond 2018. Therefore, the Pointe Scientific manufactured, we turned to their manufactured method for a G6PD reagent set, hemoglobin reagents, and hemoglobin standard that was FDA cleared for use in the clinical laboratory. Although when we reviewed all the package inserts, the published method involved manual reagent mixing and pre-lysing the whole blood specimens prior to automated analysis. So taking a closer look at this G6PD enzymatic assay, G6PD catalyzes the first step in the pentose phosphate shunt, oxidizing G6PD to 6-phosphoglucanate. And that's going to be noted as 6PG in that diagram. Now, nicotinamide adenine dinucleotide phosphate, NADP, is reduced by G6PD in the presence of G6P to NADPH. Now, that rate of formation of NADPH is proportional to the G6PD activity, and that's what we're going to measure within our instrumentation, and that's done in an absorbance of 340 nanometers. Okay. Now, this slide represents the manual procedure necessary to perform the hemoglobin as well as G6PD assay. Now, I'd like to just bring your attention to the bold aspects of it and to provide an idea of the number of minutes required to complete the task. And it should also be noted that a tech would have to devote all of their time and energy to this process, and the potential tech variability and human error associated with manual procedures as well. Now, several calculations are also required, and those would need to be performed manually without an automated procedure. Again, introducing potential errors and requiring committed techs to complete. Now, in addition to the G6PD assay, intermediate and deficient G6PD specimens must be routed for further analysis relative to hemoglobin concentration. This secondary assay is also labor-intensive and time-consuming to sort specimens and complete the manual procedure. Now, this slide depicts the manual hemoglobin procedure to illustrate again the time and challenges associated in order to process specimens in a clinical laboratory. Now, this slide here, this is given our projected annual workload. I calculated based upon the 120,000 specimens annual clinical versus soldier readiness processing specimens that are sent to us from a variety of areas. Utilizing the package inserts, this would take approximately 40 minutes of active medical laboratory scientist assay time. And you can see here from the calculation that that would require 38 full-time employees, or FTEs, per year. And given an average salary, you could see this could cost upwards of $2.3 million in labor cost alone. So considering labor constraints as well as funding constraints, this clearly was not an option for us. So we sought to utilize our Abbott A3600 automated track system to streamline the process and diminish cost. These are some pictures of our laboratory at Eisenhower. For those of you with track systems, you know they're powerful tools to yield high throughput efficiency. Okay, so I brainstormed with our stellar Eisenhower laboratory team, and we knew what our ultimate automation strategy should entail. How could we really capitalize upon that track system? We wanted to directly load whole blood specimens into the input-output module. Could we load the manual reagents into the carousels? How could we capitalize upon the automated dilution capability for onboard lysing of the erythrocytes? And could we utilize the weight area and track rules for the G6PD and hemoglobin assays? We looked at what our controls and calibrators, how are we going to set up our middleware called Instrument Manager, as well as how would we leverage the laboratory information system to automatically complete the calculations. Okay, and we wanted to take into account wash methods. We were certainly concerned about the potential for whole blood contamination. So this slide outlines our automation strategy that we employed. We utilized this to optimize our workload, diminish overhead, and improve turnaround times. Our whole blood specimens are shipped to us, so we have limited time to process. In order to streamline the implementation of this method involving both G6PD and hemoglobin assays, we sought to capitalize on that Instrument Manager middleware. The IM rules were developed in a manner that we route the specimen to the analyzer to aliquot a specimen, and then it goes back to the weight area to determine whether or not it's intermediate or deficient, and then it'll be routed back to the analyzer for the hemoglobin assay, and then routed to the storage module. And then we utilize our LIS to calculate the G6PD relative to hemoglobin, but you could also utilize your middleware or something of that nature. Now, after the preliminary G6PD activities determined, as I said, we route the specimen to the weight area, and then we perform the secondary hemoglobin assay, and then these are the calculations that we utilize to determine the G6PD per gram of hemoglobin. Again, all this is outlined in our publication as well. Okay, so this is table 1 from the manuscript, and it provides all of the assay parameters that we optimized in order to modify this assay for use in the automated platform. This can all be programmed directly into a C8000 for immediate implementation, and it'll also give individuals an idea of what needs to be modified if you're utilizing a different platform. The method comparison and precision studies are outlined in the publication as well. As shown in Table 2, comparison of both the manual and automated Pointe scientific methods yielded correlation coefficients of 0.9666 and 0.9978 respectively. And as shown in Table 3, precision studies utilizing deficient, intermediate, and normal G6PD modalities yielded a percent coefficient of variation of 0.8, 0.8, and 0.7% respectively for our patient specimens, and 1.5, 3.1, and 0.8% respectively for the control materials. We were fairly pleased by these results. The pre-lyse method corresponded to a less favorable 9.2, 2.8, and 2.5% coefficient of variation for deficient, intermediate, and normal G6PD modalities, respectively, for within-day precision. So as you can see, the data was more precise on the automated method versus the manual method, which again should come to no surprise given the variability associated with manual methods. Now, as shown in Figure 1, the linearity of G6PD was analyzed within the measured range, and from 32.5 to 14.5 milligrams per deciliter, with an R squared of 0.9985. Now, from a regulatory perspective, the College of American Pathologists requirements for modifying FDA-cleared test methods were considered along with quality assurance requirements. The following CAP checklist items should be of particular interest and should be considered in the context of all applicable checklist requirements, and I have those listed here. Now, in particular, the laboratory must maintain a list of modified FDA-cleared assays, and since this method is a modification from the manufacturer's package insert, each deviation must be clearly delineated in your standard operating procedure and approved by the laboratory director. In addition, analytical validation studies are required by CAP for modified FDA-cleared tests to ensure reliable results. For qualitative interpretation, CAP requires the laboratory to verify the appropriate method specifications that are clinically relevant. And in this case, your quality control material and proficiency test can be utilized to verify the qualitative G6PD results as well: normal, intermediate, deficient, things of that nature. So in closing, this method is available for immediate implementation utilizing the Abbott Architect C8000, which allows the technician to load whole blood specimens directly onto that A3600 track. And then all they have to do is subsequently review and certify the G6PD status, normal, intermediate, deficient, and the G6PD activity per gram of hemoglobin within the laboratory information system. That tech doesn't have to complete any pre-analytical steps or retrieve specimens or prepare any hemoglobin, any specimens for the reflex hemoglobin testing. Now, given the projected annual workload of 120,000 specimens, in conjunction with that 40 minutes of pre-analytical specimen retrieval and reflex testing task, this streamlined process resulted in a cost savings that was greater than $2.3 million. Now, what we capitalized upon was the onboard dilution capabilities on that C8000. So instead of utilizing a diluent, we replaced it with reagents. And then we were able to, in essence, utilize that diluent capability to add the different reagents into the reaction flask. So this can be implemented on other automated platforms as well. And this will allow you to streamline all of your pre-analytical steps. So again, all of the details are outlined in our publication. You have my contact information if you have any additional questions beyond the scope of the webinar. Thank you so much for your time and effort. This concludes the webinar, and I'd like to turn it over to our narrator regarding the closing action items.
This concludes today's webinar. To obtain your free Accent Continuing Education credit, please fill out the evaluation form through the link shown on the screen now. At this time, we will spend 10 minutes for a Q&A session.